Okay, so now we know how to name our epithelial tissue. We have a general sense of what the function is for each type, and we know sort of, where these tissues are going to be found. Again, I'm not looking for you to know every single location, just the main ones that we kind of touch on again. Okay, so now I'm going to be giving imagery to kind of go along with your naming of the epithelial tissue. So I'm going to go in order just as I presented them on the previous slide. Uh, and so you can keep your notes kind of, straight as well. All right. So first row here, this image as well as this is showing simple squamous epithelial tissue. Okay. So this is a superior view. We're looking down on that single layer of cells and we're seeing the squished cells. So we can see that they're actually not squished at all. And they kind of have this, irregular shape, but still we would be able to identify this as epithelial tissue because it's compact cells. And then furthermore, we could identify it as squamous because, well, one, they're definitely not cubes. Two, they're definitely not columns. So we know that this has to be squamous epithelial tissue. Now, this is probably a more traditional view of simple squamous epithelial tissue that you'll be seeing in lecture and lab, and this is from a lateral view. Now, remember, epithelial tissue lines the organs, okay? And the apical side is always going to be facing the body surface, the body cavity, the lumen, the free space. So, we do not care about any of this mesh down here, okay? When we are identifying epithelial tissue, we are looking to the surface and we are looking at this single layer of cell. Okay, this dark layer of squished purple cells, those are going to be your simple squamous epithelial tissue. Again, they're not cubes, they are not columns, and we can tell that it is absolutely one layer, so it has to be simple squamous. I can tell that it's one layer because I'm seeing right here this darker portion, that's the nuclei, and I see that that in that one cell is touching the basement membrane. And continuing on. Okay, so again, remember that the simple squamous epithelial tissue is typically found lining blood vessels and then also in the air sacs of the lungs. So we can then move on to our simple cuboidal epithelial tissue. Look at this. I wasn't lying to you. The cells literally look like cubes. Okay, so we can see that again, epithelial tissue is lining, and it's the apical side is always going to face the lumen or the free space. Right here, all inside this tube, that's the lumen. All right, so all of this right here that my cursor is going along is the apical surface. Below that, we can tell that this is really just one layer of cells, okay? It wouldn't be stratified, it is going to be simple. And we could look at this one down here as well, again, seeing that one single layer, so simple, and they are cubes, cuboidal epithelial tissue, okay? So here we would see the basal layer and the basement membrane, and then going here, we would see the apical layer, the apical surface, facing the free space, the lumen. So that is simple cuboidal epithelial tissue. And again, these are going to be typically found in kidney tubules, okay? So it's like a tube. Think of the garden hose, okay? Or a straw, right? Where the liquid flows through that water as you're sucking up a straw, lining the inside of that is going to be your simple cuboidal. Okay, now over on this image, we have hopefully a very clear cell shape. This is columnar because they're columns. Okay, now this is simple columnar. okay? The nuclei are all in one row. So it's not giving the appearance of stratified. So it's not pseudostratified. It is just simple columnar. Okay. Now, with simple columnar, we are going to see we're going to call it non-ciliated simple columnar. Because if you remember, we go back a little bit. Columnar can be ciliated or non-ciliated. Okay, so in this image that we're viewing, we are looking at non-ciliated simple columnar. The reason I know that is because the reason I know that it's non-ciliated is because there is no cilia at the apical surface. Instead, there's something called microvilli. Okay, microvilli right here. Now, microvilli, the purpose is to increase the surface area of the cell. Okay, when you increase the surface area, you're actually increasing the cell's ability to absorb. And that's really handy because this type of cell, this type of tissue is found in the gastrointestinal tract, is in your digestive system. So having microvilli increases absorption rates, which is fantastic because we need to absorb the stuff that we are digesting. All right, so we see microvilli. I don't see cilia, so I know it's non-ciliated simple columnar epithelial tissue. All right. Alternatively, this is ciliated simple columnar epithelial tissue. Okay, so before we talk about the cilia, let's just talk about how I know that it's simple and not pseudostratified. Once again, my nuclei are all in the same row. Whee! Okay, and I can see that all of these cells are touching the basement membrane, and I can see that they're all in contact with the apical surface as well. So I know it's simple, it's one layer, they are absolutely, without a doubt, columns, so they are going to be columnar, so simple columnar. Now, how did I know that it was cilia? Let's just compare. Look at the apical surface of this non-ciliated columnar versus this, okay? The cilia, honestly, I think it looks furry. I think it looks like a furry, fuzzy wall almost, okay? So the cilia is going to help move things across the surface of the tissue, okay? Whereas microvilli helping with absorption. Cilia just helps move things. So here we have ciliated simple columnar epithelial tissue. And these are going to be found in the respiratory tract, right? So in the respiratory tract, we have something called a mucous membrane, which is a membrane that secretes mucus. The cilia will help move the mucus, okay, through the bronchioles, bronchi, to the trachea, and then up to the back of our throat. And then we'll be able to swallow said mucus if needed. Cilia, or really ciliated simple columnar epithelial tissue, is also sometimes found in the uterine or fallopian tubes, and they will help transport the egg cell from the ovary down to the uterus. Okay, so cilia are transporting substances or structures in the body. Because again, epithelial tissue lines, so this tissue is going to be in contact with this free space. Okay, now this one. This is still cilia. Okay, so this is ciliated. And I think you can all agree with me that this looks like a columnar cell. This also looks like a columnar cell. However, if we're trying to find the nuclei and we found a nuclei and we were drawing a straight line across, are we going to hit Are we hitting all nuclei? No, we are not. So that means that we have this staggered appearance of nuclei, giving it that pseudo-stratified classification. All right, so we have cilia, making it ciliated, pseudostratified columnar epithelial tissue. Okay, the ciliated pseudostratified columnar epithelial tissue. This is going to be also found in the upper respiratory tract, again, because cilia helps move the mucus. Typically, I identify ciliated pseudostratified columnar epithelial tissue in the trachea specifically, but you could just say upper respiratory tract. I know it's a mouthful, but you have to be specific when you are naming these tissues. Oh, I will say though, in lab, you don't actually need to say ciliated or non-ciliated. I think it's important in lecture for us to be able to identify and absolutely comprehend the difference between ciliated and non-ciliated. Okay, whether it be ciliated or non-ciliated simple columnar or ciliated or non-ciliated pseudostratified columnar. Okay. Okay, so here's just a 2D image again showing the difference between microvilli and cilia. I think cilia is just more furry and the microvilli is a little bit more structured. And it's, again, the functionality, microvilli helping with absorption, cilia helping with transport. And so you can also see the difference between, okay, here's the simple columnar, pseudostratified columnar. Remember, if it doesn't have cilia and instead it has microvilli, we call it non-ciliated. Okay, so moving on to our our stratified epithelial tissue, meaning that it has multiple layers. What we have here are the stratified squamous epithelial tissue. There are two different types of stratified squamous epithelial tissue. Okay, now there's non-keratinized and keratinized. Non-keratinized is going to be found in places like the esophagus and the vagina or the rectum. Non-keratinized. Keratinized will be found in the epidermis of the skin. Okay, both of these are stratified squamous, but we just have these different classifications. Now, keratin is a protein, and its main job is to protect the skin and the underlying tissues of the skin as well, right? So, non-keratinized stratified squamous epithelial tissue doesn't have a lot of keratin, and it's really going to be found in places where there's a lot of moisture and a lot of mucus, okay? Some type of mucosous glands, all right? So non-keratinized, again, esophagus, rectum, vagina, really moistened, dark areas, not a lot of keratin, okay? Keratinized stratified squamous epithelial tissue, again, has a lot of keratin and, again, is found in the skin. All right, so if we just kind of understand what keratin means, we can kind of help us find the locations. Now, the easiest way to identify this type of tissue is to look at the apical surface. Okay, now, when you're looking at stratified epithelial tissue, it's not that all of the cells are going to be the same shape, okay? Because this tissue goes all the way down to this wavy purple layer. Okay, and this tissue goes all the way down to this darker wavy layer. All right. When you get down to these lower layers, we're not really seeing squamous anymore. So we're always identifying the cell shape closer to the apical surface. All right. But again, looking at the apical surface, we can see that these cells are kind of dying off. Okay. We have these dying off keratinized or non keratinized cells. And it's the same or maybe even worse over here with the keratinized. This loose and flaky layer that you're seeing, that is the top layer of your skin. That is the stratum corneum. That is your dead skin cells, essentially, that kind of flake off. Okay, so if you had an itchy spot on your arm and you were scratching it and you had some skin cells that were flaking off, that's what they would look like under the microscope. Okay, so keratinized, it's in the skin and you can identify it because it has that dead skin layer. All right, over in the non keratinized, you can see that they're kind of squished and kind of shriveling up a little bit, but it's not as dramatic. All right. So that's stratified squamous epithelial tissue. The only other one that we're going to look at and identify is the transitional epithelial tissue. All right, so remember the transitional epithelial tissue is weird because it has the transitional cells. If we remember, transitional epithelial tissue is found in the urinary system, the urinary bladder. Urinary bladder constantly stretching and then resting, right? Going back to normal. So, at the apical surface, the cells, when resting, are going to be more cuboidal shape. Okay, but then when the bladder expands, the urinary bladder expands, these cells at the apical surface are going to stretch and they will take on a more squamous shape. And that's transitional epithelial tissue. Let's talk about glands. Okay, so we know that the functions of epithelial tissue include glandular secretions. Now, what is glandular epithelial tissue? It's literally epithelial tissue that has glands. Okay, so hopefully we've heard of a gland before. There are two different types of glands. Exocrine and endocrine. And I have a long side-by-side comparison of exocrine and endocrine glands. We don't really need to go too in depth on that because that is more so a gen bio concept. But essentially, a gland, regardless of the type, is going to secrete substances into a duct or onto the surface of the body or into the blood. Okay, so a really big difference here with the two types of glands, exocrine and endocrine, is endocrine doesn't have ducts. It's secreting hormones directly into the bloodstream. Exocrine glands, these guys actually have ducts, okay? So they are going to be releasing whatever secretion, whether it be oil, wax, sweat, milk, whatever it's going to be secreted through a duct. Okay. Now, in the exocrine glands, there's two different types of classifications. We have the unicellular and the multicellular. All right, so unicellular, the only example that we have here is a goblet cell. We're not going to go too in depth here, okay? And again, you don't need to know every single portion on this side-by-side comparison. It's really just there in case you don't remember from the Gen Bios. What I do want to point out are the multicellular exocrine glands. So you don't need to know all of these. You don't even really need to know any of them, but I want to point out some of them because they will come up time and time again. Okay, So I'm going to look at C first. And so we have this coiled shape. That is a sweat gland, okay? An eccrine sweat gland specifically. So those sweat glands are going to be coiled in their shape. Then we have sebaceous glands. That's your oil gland. That's going to be over here at E, these branched multicellular glands. All right. And then mammary glands, those are going to be down here at G. We have this compound branching of the multicellular glands. So I just kind of want to put a visual there for you of the different types of glands, especially when we get into the integumentary system. It may be easier for you to identify these types of glands after having seen these. Maybe. Again, not testing you on this. Okay? When we're looking at the glands, we got to talk about the exocrine gland functions. Okay? So how these guys are actually working. Now, we're really only paying attention to the multicellular, right? We don't care about endocrine because that's really for the endocrine system. So we're just looking at the exocrine glands, and we're looking at the 3 different methods of secretion. So the first one is called the merocrine secretion. The gland that they have here, for an example, is salivary, but it could also be sweat glands. Sweat glands are going to perform merocrine secretion. What happens with merocrine secretion is essentially exocytosis. So if we remember what exocytosis is from Gen Bio, it's where we have something that gets packaged into a vesicle. That vesicle fuses with the cell membrane, and then it releases its product. Okay, that's merocrine secretion. That'll be done by salivary glands, but more so what we're focusing on are the sweat glands. The second type of functional classification of exocrine glands is apocrine secretion. Now, this gets a little tricky because there is a sweat gland that is called the apocrine sweat gland. Guess what? It does not do apocrine secretion. All sweat glands perform merocrine secretion. All sweat glands, merocrine. Okay. They called it the apocrine sweat gland because they thought they did apocrine secretion. They don't. Okay. Mammary glands, however, do apocrine secretion. Apocrine secretion is essentially the same thing. You package the substance into a vesicle, but instead of the vesicle fusing with the cell membrane, it actually just, a portion of the cell gets pinched off and is then part of the secretion. Okay. And then we have holocrine secretion, which we'll see with our sebaceous glands. Again, those are the oil glands. And they are going to, you'll see that the secretion will get packaged up, and then the cell actually dies and gets secreted with it, essentially. Okay, cell division does happen and the cell gets replaced. Yay.